Thank you very much for the introduction. I'm very happy to contribute to this. And you know that the entire world is looking at the struggle that you're going through. And I have all sympathy for seeing you struggle while your life is being disrupted and your studies are being disrupted. So I would like to tell you about my life in science. And for those who are not familiar with my work, there will be slides where I explain it, but it's somewhere in the middle. So I'm just going to go into my screen. Okay, so my life in science really went from physics to biology to chemistry, and I'll explain this. I was educated as a physicist, and then in the end, I got a Nobel Prize in chemistry. And I'm showing this path in a zigzag because there were so many unexpected events. I mean, I could not have dreamt to go from here to here. Now, the English language has a term which comes from the Greek, peripatetic, which means traveling from place to place, in particular working or based in various places for relatively short periods of time, nomadic, itinerant, traveling, wandering. So this is when I look back, this is exactly how my life seems to me now. And I just wanted to give you a tribute. There's a statue just two blocks from here, from my apartment in Manhattan on the Upper West Side. And a few days after the war started, somebody planted a bouquet of flowers pointing clearly to Ukraine. And this bouquet is still there in place. Now, I was born in Siegen. The Siegerland has been for centuries a center for iron mining and manufacturing. And this is me with my family. I was just born in 1940, so this was in World War II. And I grew up in my parents' house, Engsbachstraße 3 in Siegen. It's a very stately house, very large. And in 1944, when I was 3.5 years old, Siegen was 80% destroyed. My parents' house, fortunately, was only hit by an incendiary bomb, and this essentially finished the roof, but the rest of the house remained there. So we had to leave for 1 year and stay at somebody else's apartment. So in that house that I showed before, there was this little place under the veranda, which we called Kabäuschen. And when I was young, and I remember I was 7 or 8 years old, I started science experiments here. I did all kinds of things. I didn't know that it was science. I had little bottles filled with stuff and mixed them together. I heated coal and so forth, and watched a gas come out, and I was able to lit the gas. So it was very exciting, but I couldn't share this with anybody else. Here I am in biology class in high school, 1957. And we had a very inspiring physics teacher, Wilhelm Schröder. And here he is shown. We gave our teachers a mock exam at our Abitur fire. And so he finished successfully to put a light circuits together. And remarkably, we had a very small class of 20 and 4 students became physicists. Here I'm in Freiburg in Germany at the university. And this is where I did my 4 diploma. I'm not sure whether you have the same kind of system in Ukraine, but 4 diploma, PhD, and so forth. So this is the academic ladder. So I got my 4 diploma in physics from the university in Freiburg. And then I went to Munich and stayed there for several years. And in Munich, I was at the university in the Physics Institute and did my diploma in experimental physics. Then after my diploma, I looked for a place where I could continue my graduate studies. And I wound up in the lab of Walter Hoppe. So this was a very unique lab. First of all, I wound up there because I developed an interest in electron microscopy during my diploma thesis. So I was looking within Munich, I was looking for a place where I could continue or actually get into electron microscopy in a serious way. And my mentor, Walter Hoppe, was a very unique person because he was an X-ray crystallographer who then in mid-career turned his interest to electron microscopy in the early 1960s. And so I did my PhD thesis at his institute and finished it in 1970. And the theme of this was all optical diffraction, digital correlation analysis of electron micrographs, contrast transfer function correction, and drift as manifest in Fourier space. So these were all things that had not been explored before. And I was one of the first people who did image processing to examine electron micrographs. And so this is the institute, which is just a few blocks from the Oktoberfest. In fact, X-ray and electron microscopy already at that time were very closely related. In fact, electron microscopy picked up a lot of concepts from X-ray crystallography. And my mentor, Walter Hoppe, had a very close relationship with Max Perutz, who was one of the pioneers in protein X-ray crystallography. So they co-organized meetings, workshops in Hirschegg and in Alpbach. So my first meeting, my first scientific meeting was in 1968, and it brought X-ray crystallographers together with early pioneers in electron microscopy. And if you know anything about the field, then the name Harold Erickson, Richard Henderson, Ken Holmes, Hugh Huxley, Nigel Unwin, maybe you have heard of them. So I already met all these people at that time. And to give you an idea about what it meant to do image processing at that time, this was a very advanced machine that I did my work with. It had a memory of 4K of 16-bit words, and was introduced in 1965. So it was introduced just a couple of years before I started my work there. So this is quite a challenge to process images with this kind of equipment. Thousands of these of the computer power or maybe hundreds of thousands of this computer power fits into a current cell phone. So this is where I did my thesis. It was an investigation of high-resolution electron micrographs using image difference and reconstruction method. This was later published in 2019, much later, just because the Technical University wanted to honor me after my Nobel Prize. And so I have a very nicely printed edition of this now. And then came my Wanderjahre. I got a Harkness Fellowship, a very prestigious fellowship from 1970 to 1972. And in the course of which I went to the Jet Propulsion Lab, to Caltech in Pasadena, California, and then to the University of California in Berkeley, and to Cornell University, Ithaca, New York. Now, I show Bob Glaser here because Bob Glaser was my mentor during the short time that I was at the University of California at Berkeley. Bob Glaser is really one of the true pioneers of cryo-electron microscopy. He tried this technique very early, but at the time it did not take off, and it was not. There were still improvements necessary in order to make it work. But these Wanderjahre were very influential for me later on. And I also connected with a lot of people that I later had a very close relationship with. And then I went briefly back to my initial institute, but meanwhile, this had moved to Martin Street and became part of the very large complex of the Max Planck Institute for Biochemistry. But I only spent a very short time there. And then I got an offer to become a group leader at a Cavendish Lab, Free School Lane in Cambridge in England. Now, this is a very prestigious lab that has a lot of background in discoveries and invention in physics. And here's a plaque that celebrates this outside. And you will probably know the name of Ernest Rutherford. And he discovered the nucleus in in 1919, he was appointed as a Cavendish professor. And very interesting, in the courtyard of the institute that I was working in at the Cavendish, there was the barracks of the Laboratory of Molecular Biology. So the Laboratory of Molecular Biology that is now famous at the MRC had its origin in barracks in the courtyard in the Cavendish Center, Cavendish Lab. So this lab later moved to Attenborough's Hospital and to become part of the MRC. So the very first stunning achievements of the Molecular Biology Institute came right from this place. And so Max Perutz, I already mentioned him before, together with his student John Kendrew, got a Nobel Prize for solving the structure of hemoglobin. So this was the first, one of the first protein structures that was solved by X-ray crystallography. And then I made a big jump in my career. I got a job offer from the Wadsworth Center at the New York State Department of Health as a senior research scientist. And for some reason, even though I was so young in my career, I was known for digital image analysis of electron micrographs. So I got an offer of a position as a group leader where I would use image processing in support of the high voltage microscope. So the person who brought me there was Don Parsons, who had this vision that using high enough voltages, in this case is 1.2 million volt, one could penetrate very thick specimens, and when one could find viruses that were associated with cancer. He believed all cancers were caused by viruses, which turned out to be wrong, but it was part of his his conviction that he convinced the governor at the time to put his money into this very large equipment. So I'll just tell you something about my research in a few slides. Because most of the ideas that were later, that I was acknowledged for, came all in at the Wadsworth Center while I was working there. It was a very quiet working environment. I didn't have to worry about grants. I had plenty of support from the state government, initially at least. So it was a very nice time to really develop ideas and put them in practice. So the whole context is molecular machines in the cell. We have many large molecules that form allegiances with other molecules, so they go through entire work cycles, and they work in a processive way. And in order to understand the processes in the cell, we wish to know the structures of all components, but also the way they interact dynamically. And reductionism is a concept where we study a subsystem in isolation, in vitro, which means we extract it from the cell and then hoping to at least approximate the processes in the environment of the cell. Because in the cell, everything is packed together very tightly, and it's very difficult to look at any component with high resolution. But that is possible if we take it out of the cell and then look at it separately. So in 2017, I got the Nobel Prize along with two other people for developing cryo-electron microscopy for the high resolution structure determination of biomolecules in solution. So the important thing is in solution, okay? So they're not in the context of the cell because it's too difficult. So here you see a cell, a typical cell where everything is packed together, and we somehow have to dissolve the cell and take the molecules out, have them in solution, and then study them in solution. Now, at the beginning of these efforts in electron microscopy, to get to the resolution where one can recognize molecules, there were groups, they were all at the MRC that I mentioned before, at the Laboratory of Molecular Biology, and they all looked at the at molecules, but they all looked at molecules that were aggregated into crystal form. So they never looked at molecules in isolation, never in single molecules, because they believe that by the packing of the molecules, one get really a much clearer picture. And so the pioneering achievement is by de Rozier and Klug in 1968, who applied for the first time certain computer algorithms to perform a three-dimensional image of a molecule. And this is the molecule of a bacteriophage tail, which has helical symmetry. And then somebody else, Tony Crowther, applied other or developed other kinds of programs in order to study a spherical virus. So he achieved the first such reconstruction in 1970. And then Henderson and Unwin, Richard Henderson is one of the people that I got a Nobel Prize with. Henderson and Unwin he got to a very high resolution at the time at 7 angstroms, and they got images of a helix bundle of bacterial rhodopsin. Now, what all these have in common is that molecules are studied in aggregation. They all form crystals together. And so that was bothering me, because not only did they have to develop different kinds of programs for different kinds of symmetry, but also the single molecules, molecules that could not form crystals could not be studied with these techniques. So why crystals? And so this was this flash of an idea. Single particle techniques, the structural information is obtained from images of single, which means unattached molecules that exist in many copies. Molecules are free to assume all naturally occurring conformations. Molecules are randomly oriented, and a single snapshot may already give us hundreds of particle views. And as we collect more snapshots, more orientations will be covered until we have enough for reconstructing the molecule in 3 dimensions. So this was the idea. And many, many programs had to be developed in order to make this happen. And so this was a very exciting time for 10 years until we got our first 3-dimensional reconstruction. And these are a few images from that time. You can see how primitive the computer equipment was at the time. And of course, I had the fashion, I wore the hair fashion of the time. You can see me here. So the first single particle reconstruction we obtained in 1987, but that was not with cryo methods, but with negative staining, which was very much inferior, but this was the only thing that was available at the time. And I'm highlighting my postdoc, Michael Rademacher, who developed many of these programs that finally made this kind of approach happen. And this was really the first clear images of the ribosome that we obtained at the time. And this was a very big splash, and it was a very big milestone. And here, the 50S ribosomal subunit is presented as a contour stack. This is a very heavy box where individual transparencies are mounted. This was still in my lab. And then when I got the Nobel Prize, I brought it to Sweden, to Stockholm, where it is now on a permanent exhibit in the museum. So in essence, we can now take molecules from solution, go through all kinds of software, and then wind up with a beautiful high-resolution image. And so nowadays one can do this, and this is our highest resolution image of the ribosome, 3-dimensional image of the ribosome that we obtained in 2016 with much improved cameras. I got to Columbia in 2008. So I left Albany and the Wadsworth Center in 2008, and I'm still there right now. On October 4th, 2017, I got this call at 5:18, and it was very exciting to hear a Swedish accent of the secretary on the phone, and and I had an inkling what this was about. And then I joined the press conference by phone. That was immediately like 3/4 of an hour later. It was a lot of excitement. And here, reporters came already at 7 o'clock, and we had to pose with our dog, Daisy. And Daisy, incidentally, I took a picture a week before I got this call. I took a picture in Central Park with Daisy. And because of very stunning effect of the 3 dimensions and the projection. And so it is sort of epitomizes the whole approach that I brought in. So in Stockholm, December 2017, this was really an absolutely glorious event. And I show you a few pictures of it to give you an idea of what a spectacle this is. And here I am with this box and presented along with Jacques Dubochet and Richard Henderson. Jacques Dubochet shows this instrument for plunge freezing, a very primitive device. And Richard Henderson shows off an atomic model of the bacterial rhodopsin. Okay, so here we go through explanations of this in the Nobel Museum. And then they have this very funny tradition in the Nobel Museum. There is a cafeteria, and the new Nobel laureates are supposed to sign the bottom of each chair. So this is my contribution here. So if you ever wind up in that cafeteria, just turn the chairs around and look for my signature. And here we are. Here is the rehearsal, and that's why we are so informally dressed. And here is the big event. Here we are all together. And this is with my wife, Carol, who has been incredibly supportive all my life. Signing the book, big book. If you look back and forth, you find people like Obama and many people of note that we know from science and politics. And this is such a glamorous affair in the Rathaus in Stockholm. So I have just a few slides, a few additional slides to emphasize that cryo-electron microscopy, the single particle kind of cryo-electron microscopy, has been instrumental in giving us details on the interaction of the COVID-19 virus with the host. So it's been incredibly useful in order to even develop the vaccines, to show the stabilization of the spike protein by introducing a certain proline mutation. So this is not only true for the COVID virus, but for many other viruses that have caused recent pandemics, such as dengue and MERS and so forth. And so you will find many of these kinds of publications. And if you go to Google and put in cryo-electron microscopy, you get hundreds and hundreds of pages like this, where you see these structures that have been solved with this methodology. So in conclusion, cryo-electron microscopy of the single particle kind has opened a new era in structural biology and molecular medicine. There's no need for crystals. Very small sample quantity is needed. Resolutions in the 3 to 4 angstrom range is now routinely available. Multiple structures can be retrieved from the same sample, which gives us clues for function. And molecules are enclosed to native conditions, which cannot be said of molecules that are studied in crystals. And solving structures of membrane proteins particularly is much easier than with X-ray crystallography. And all in all, we have experienced a huge expansion of structural database relevant for molecular medicine. You can see the rise of depositions from cryo-EM here, the yellow line. It really goes steep up, whereas the other depositions are simply leveling depositions from X-ray crystallography and NMR spectroscopy. Now, you're welcome to go to my Frank lab. You might be able to simply type in Frank lab and it leads you to this. And there are many useful information and you see all the publications, but I just wanted to draw your interest to this icon here, which is Nobel 360. This is an initiative of Lindau, of the Lindau Foundation, Lindau Nobel Foundation, where they send people to different parts of the world into labs of Nobel laureates, and they make documentation, 360-degree kinds of documentation of the lab environment. And so you find something like this, but you can now steer around and go look at all the different features. Here I'm standing in front of the high-end electron microscope, Titan, and here I'm standing at a place, a very historic place, not very far away from our lab. It's a little lighthouse and a George Washington Bridge. There is a children's story, I think that was published in the 40s, that depicts the little lighthouse as a person, and the George Washington Bridge as a sort of a big brother that has joined it. Okay, so with that, I'd like to thank you for your attention. And this is a picture on the roof of our lab, of the building of our lab, and we are right now we're looking toward the south. The camera looks at the south. We're all looking in the north, and I'm so proudly displaying the Nobel poster. This was taken in the summer of 2018. Okay, thank you.
Thank you very much, dear professor. We have many questions to you. So first of all, we have many similar questions. So do you expect further improvements in microscopy? What kind of improvements it can be, for example, in 100 years?
Oh, 100 years. Oh my God. I don't want to think about 100 years from now. But the progress in science is very rapid. The progress in instrumentation is very rapid. So right now I'm involved in some developments which really go out of simply taking three-dimensional static pictures. I'm looking at the motions of molecules, and there is such a thing as a time-resolved cryo-EM, and this is a method that I've been developing in my lab, and we have very good results already. We've published them in Nature, And then there is another very far-reaching development, which is that if you take very large data sets, like millions of molecule images, then you can reconstruct the free energy landscape of a molecule, and that gives you clues to function. So these are two developments that really have nothing to do with getting to higher resolution. Now, higher resolution has been achieved already, and the best resolution is even better than 1 angstrom so far. So any development further is really pointless. You don't need that kind of resolution for drug developments, for instance.
Okay, the next question is from Tatiana. You mentioned that the study of samples takes place in a liquid medium. Is it water or is it still a specific solvent?
No, it's water. It needs to be water. So the molecules are brought in solution onto the grid, and then using blotting, this water layer is made very thin, and it needs to be very thin because electrons with 300 kilovolts cannot penetrate very far. So this is, and then the grid is plunged very quickly into liquid ethane, which is on liquid nitrogen temperature.
Okay, the next question is from Yuri. Is it essential for a living cell to be a 3-dimensional? So could a planar cell act like a 3D one?
Our cell, well, you know, I mean, it's a matter of degrees. Everything is 3-dimensional in this world, and you can say, you know, can a cell be very flat? And certainly, many cells can be very thin and extended, but they are always 3-dimensional. You know, molecules are 3-dimensional, and the cell consists of molecules.
Okay, now we can move to some motivational questions, I mean, about doing science overall. So what inspires you to make science? Why did you become a scientist?
Well, I try to explain. It was somehow an innate curiosity that developed very early. So with 8 years old, I already tinkered around with all kinds of things, with the things that are found outside, and little bottles. I simply experimented at a very early age, and so physics immediately attracted me in high school, and I excelled in physics, and there was no question that I would study physics later on.
Okay, now we have some urgent question due to our challenges. How did you overcome challenges when you face it? How do you usually do not give up? What do you usually do to not give up?
Well, I think, you know, my idea was very unconventional, and it was very difficult to convince other people. We couldn't get our papers published and so forth, and it was very important that I had some people who believed in this, and so I could interact with them. I also always had support by my wife, which is very important. So I went through stretches, very long stretches of big disappointment, but then I had this idea that I would really prevail, and then also if a very good recipe is if you have you shouldn't have a single project. You should have several, and if you get into a dead end with one, you switch over to another one, and so forth. So there's always during a time that you pursue this project number 2, something happened with your brain, something cooking there, or there are new influences that you don't even recognize, but they somehow slowly bring you further in project number 1.
Okay, the next question is, what is the most important item to become a successful scientist, in your opinion?
Well, curiosity, and then I think the ability to take advantage of what I call peripheral vision. You make many observations that you might otherwise discount, but it is very important, in my experience, to really take in whatever comes, even if it looks unrelated to what you're pursuing. But there are always ideas that come from the wayside.
Okay, and maybe I will ask the last question, because we have a lot of questions actually, but we have a strict limitation of time now. So we have traditional question. I ask this question to all our speakers. What pieces of advice can you provide to young scientists, to school students who just start to do some research and some science?
Well, so there are two things. One is what I just told you, peripheral vision. Keep your eyes open to things that sort of come from the periphery, that seem at first completely unrelated to what you pursue. But my experience is that there are always clues. The other one is stubbornness, because I developed my ideas in contradistinction to my mentor. I thought the way he approached three-dimensional reconstruction of molecules was really not productive, and I was very persistent in this. So that's my advice. Your mentor might be wrong. Okay, it's maybe it's a very very difficult thing for mentors to hear, but you really have to come on your own with your ideas and sort them out by using all resources that are available, and not just listen to just one person.
Okay, thank you very much for joining our lecture. Thank you again for your time and pieces of advice. Шановні учасники, всім дякую, хто під'єдналися до нашої лекції. Нагадую, завтра о 16:00 на вас очікує чергова лекція від Нобелівського лауреата з хімії. Тому реєструйтесь на нашому сайті і отримайте завтра посилання на завтрашню лекцію. Thank you very much again and have a nice day. I hope to see you in Kyiv with physical lecture someday.